and turn that into stuff and not use CO2. And the problem is that doesn't get the plant any sugar, which it needs to survive. So Rubisco is not actually very good at its job. And so there's some solutions that plants have come up with. So most plants are called C3 plants. Okay. The average plant is a C3 plant. The plants that have kind of adapted and come up with solutions to this issue with Rubisco are called C4 or CAM plants. Those are the two options. Okay. So C4 is like they've surpassed C3. They're doing a better job. Okay. Sophia, what is the plant pictured under C4? Do you know what that is?
Uh, no.
No? Okay. I used to have a job where I picked that.
Is it wheat?
That's a good guess. No, but wheat I think actually is another C4 plant. It's corn. That's what corn looks like it before you pick it off. There's some corn you can see at the bottom. Yeah, so corn is a C4 plant. And then the CAM plant, so CAM stands for crassulation. Oh, now I've just crassulation acid metabolism. And it's found most often in succulents or cacti. So that's what I pictured on the right there. Okay. So basically, I mean, this is a bit of a complicated diagram. We don't need to go in detail about this. What C4 does, I mean, I can just kind of, I think we can kind of skip to, I'm wondering what's the best way to do this. Okay. So essentially the issue is if you are a plant and you're trying to get water, right? So to get water, if this is your leaf, there's gonna be a lot of really bad drawings here. This is your leaf, and there's these openings. Emily, do you remember what the openings are called?
Tomato.
There you go. Beautiful. Stoma, stomata. So these openings in your leaf, you got the water leaving. Yay. Okay. The problem is when the water is leaving, there's a hole. And so the oxygen, the oxygen it can also leave. We're good. Okay. So that's fine. We're happy with that. The problem is that if it's really hot and dry, which it is a lot of places, and it's really hot, you don't want your stoma open or your stomata, sorry, open because your water will leave and then you will get dehydrated as a plant. So you want to keep your stomata closed. But if you keep your stomata closed, then the water can't leave. That's good. But the oxygen is also trapped inside the cells. And if the oxygen is trapped inside, all of a sudden the amount of oxygen goes up. Okay. And that's a problem because we need to have more CO2 than oxygen. Otherwise, the Rubisco will use the oxygen. Okay. So we want the enzyme using this. So we need to make sure that there's more CO2 than oxygen. And so plants have come up with these two strategies. So the first one is the C4 plants, and C4 plants have decided to use two locations. So what they do is they have two reactions that'll take place. So they'll have this extra cell, basically. And so in the mesophyll cell, what they'll do is they'll take in CO2 and they'll kind of concentrate it. So they'll store it up. So there's a lot of CO2. Okay. So they store it up in this molecule called malate. Emily, do you know what the 4C next to malate means?
Oh no.
Okay, so it means that there's four carbons in it. Okay. So malate's a four carbon compound. So we're gonna like increase the amount of carbons here, store it up. And then what we're gonna do is the actual Calvin cycle is gonna take place in this bundle sheath cell. So we're gonna concentrate the CO2 in this malate compound. Then we're gonna send it into the bundle sheath cell. And so that means that the amount of CO2 in the bundle sheath cell is always going to be higher and we'll kind of keep the oxygen away. Okay. So basically what we're doing is we're increasing the amount of CO2 so that there's always more CO2 than oxygen. And we do that using a separate location. Okay. Now, in a CAM plant, they do something similar, but instead of using different locations, they use different times. So in a CAM plant, what they do is at night, they'll open their stomata and they will like because it's cooler. And what they'll do is they'll concentrate their CO2 at night. And then during the day, they can close their stomata. They've got a lot of CO2 built up. So they've got more CO2 than oxygen. and then they send it into the Calvin cycle during the day. And that way they can make sure that the Rubisco is using CO2 and not using oxygen. Okay, does that make sense to everyone? You can say no, that's fine. I'll just re-explain stuff. So essentially C3 plants, these are our normal plants. The average plant is called C3. C3 plants, just CO2, Calvin cycle, sugar. C4 plants are going to do concentrate our CO2 in a different cell and then send it into so they have two locations basically. They'll concentrate it in one cell and then move it to a different cell for the Calvin cycle. And CAM plants, instead of doing a location, they're going to concentrate their CO2 at night and then they'll do the Calvin cycle during the day. Okay, so these are basically three different strategies to get past the whole drying out issue, which happens because it's hot and dry some places. Okay. Okay, so overall, this is what you guys learned about. So we got our photosynthesis. The purpose of photosynthesis is to make sugar. And so it uses, what kind of energy does it powers photosynthesis, Kathy?
Solar,
Sorry?
like the sun.
Yeah, sunlight, sun energy, photons. So it's powered by light and it uses that light and it uses water. And ultimately, we've got the light reactions and the dark reactions, or the light reactions and the Calvin cycle. And using both of those together, we can make sugar. And that's really handy because then we can eat plants and get food from them. Does anyone know Calvin's first name? Calvin's his last name. Sounds like a first name. His last name is Calvin. His first name was Melvin. So his parents named him Melvin Calvin, which is why I think he ended up becoming a famous scientist because, what are you going to do? Okay, so any last questions about photosynthesis before we move on? No? Okay. So our next unit is on cell communication. So cell communication is important because we are made of cells, right? We are multicellular organisms. I've now forgotten who I was at. Who did I ask last? Sophia, did I ask you last?
No, you asked Kathy.
Okay, it is your turn then. Okay, so Sophia, do you know if we are made of eukaryotic or prokaryotic cells?
I don't know.
Okay, so we're made of eukaryotic cells. The way that I remember that, so it's spelled like this. So there's these two words. There's every living thing on earth is either a eukaryote or a prokaryote. The way that I remember that we are eukaryotes is it sounds like you carry it. You have to come up with a lot of strategies to remember some biology. So eukaryote, as in like you are made of eukaryote cells. So all animals and plants, everything multicellular on earth is a eukaryote. And then single cell things like bacteria, a lot of them are prokaryote cells. So that's going to come back again. So cell communication, we are made of cells. We are made of eukaryotic cells and they need to communicate, but even cells living on their own, like bacteria, also need to communicate. Okay, so technically, actually, I don't love your textbook for this, so that's why. So technically, there's three main categories of communication if you're a cell. So the first one, which is kind of strange, is it's called autocrine. Okay, so autocrine is when a cell communicates with itself. So that's almost like if you're talking to yourself or if you're writing notes for yourself, something like that. So the cell will actually like release little chemicals and then the chemicals will bind to the cell and the cell will be telling itself something. And that's because cells don't have brains, so they can't think things through. Instead, what do we consider the brain of the cell, Emily? Which organelle?
I don't know if it's right, but the mitochondria.
Ooh, that's a very good guess. The mitochondria is, they call it the powerhouse of the cell, which I find very irritating. I would consider it more like it's like the battery center or like it powers the cell. But the brain of the cell would be the nucleus where all the DNA is. But it can't really think. So it's got all the information, but it can't make decisions. So sometimes the cell just has to release stuff and talk to itself. So that's the first one, autocrine. The next one is paracrine, and paracrine communication is when you're communicating with a cell nearby. Okay, so let's say you have two muscle cells near each other, they might send each other information, just release it. So the do you know, Kathy, do you know what the liquid outside of a cell is called? I'll give you a hint. The fluid inside a cell is called intracellular fluid. What do you think the fluid outside of a cell is called?
Extracellular.
There you go. Perfect. That's one thing I like about biology. It's pretty logical. So outside a cell's extracellular fluid. So we technically, our cells, there's fluid around them and in them. We're full of liquid. And so they would just release directly. And then the last main kind of communication is called endocrine communication. And that's when you have cells really far away. And so they're going to communicate using blood vessels. So stuff will be sent in blood vessels. For example, if your brain wanted to communicate, well, actually, you could use nerves too. But anyways, endocrine is mostly in blood vessels. So if your brain is sending signal to a muscle or something, they might send stuff in the nerves and then it releases in the blood. Or actually, a better example would be organs communicating with organs. So if your liver is trying to say something to your heart. So that's the furthest away things. Okay. So the first example they're giving here is cells that are actually connected together. Okay. So in animal cells, when they're connected together, it's called gap junctions. And in plant cells, when they're connected together, they're called plasmodesmata. Okay. Sophia, what is probably the main difference between plant and animal cells? It's in the picture. Sorry, did you answer and I missed it?
Like
You know, it's a plant cell because it has a
cell wall.
Perfect. Exactly. So they've got cell walls. So that's why the connections are a little bit different. There's a wall in the way. So if cells are actually connected, then they can just send stuff through their membranes. That's great. Emily, can you think of any cells in our body that are connected?
I don't know.
Okay, so skin. You really want all your skin cells really tightly connected so that your insides don't fall out. Okay. So that would be the easiest one. They're fully connected. When they're not, also potentially if you have, so all of our cells are covered in molecules that stick off of them. And sometimes those molecules could actually bind to other molecules on the surface of other cells. So that would be another way to communicate with really close by cells. Okay. So now it's the one, I mean, I don't love the way it's taught in this textbook, but now they're giving the example of paracrine. So that's cells that are nearby. So they're releasing stuff. Another example, which is technically kind of falls a little bit under paracrine, on the right side is synaptic signaling. So that's with your neurons or nerve cells. So it's to do with your brain. Your brain is made of neurons, so that's how they communicate. And that's how your nervous system communicates with itself and with muscles, is it releases stuff between the neurons and other cells. Okay, so that's things that are close to each other. That's pretty obvious. Here's the endocrine one I mentioned. So the classic example of endocrine is hormones. So your endocrine system is your hormone system. Who am I at? Kathy, can you name one hormone?
I don't know.
No hormones? Okay. Testosterone, estrogen, insulin's actually a hormone, progesterone. I don't remember a lot of the hormones. There's a lot, but yeah, usually the ones people think of are like what we call the sex hormones. So testosterone, estrogen are the main ones. Okay, so that's the last one to communicate. So you'd release molecules into a blood vessel and they would travel. Okay, so that's for really far away things. Okay, so how does signals kind of work? You've got one cell and it's receiving some kind of a signal, and then it's got to do something with that signal. And so there's always 3 steps to it. So your 3 steps are you're going to have your signal reception. So basically, any molecule that binds to another molecule. So you should have already done enzymes. So if you have something and it sticks into another molecule, the smaller one that's binding, we call a ligand. Okay. So that would be our signaling molecule. So it's a special shape and it's going to fit into a receptor. Okay. So similar to enzymes, you've got a signaling molecule or receptor. So that's the first one. That's the actual like receiving of the signal reception. Okay. That's always step one. The second step is signal transduction. So basically when something binds, it's going to bind in a specific location in the cell. So in this case, it's binding to what part of the cell, Emily? Is the receptor in?
The
If you want to cheat, it's labeled.
plasma membrane.
Perfect. So in the membrane on the outside of the cell. So that's where that receptor is found. So then it has to get that information from from the plasma membrane all the way somewhere inside the cell. So that's signal transduction. And there can be a whole bunch of molecules in between. Here they're giving 3. There can be like hundreds. It can get real messy. And then lastly, the cellular response. What is the cell doing? So the cell has been given information and now it has to decide what to do. And they're giving an example here. So the release of glucose from glycogen is an example of that. Okay, so those are our 3 steps. Oh, this is gross. Okay, so I once took a course called Animal Adaptations because I thought it was going to be fun and I thought it was going to be about animal adaptations. And the entire course was about G protein coupled receptors. And this is not my favorite part. Okay, so this is a protein. It's specifically called a G protein coupled receptor because it is a receptor that is coupled with a G protein. So it's very confusing naming. It's not a G protein, but it basically always is going to work with something else called a G protein. So that's why it's called a G protein coupled receptor. And this specific molecule, it has what we would call 7 transmembrane domains. Okay. Kathy, do you know what, you want to guess what a transmembrane domain is?
I don't know.
Okay, sure. Transmembrane means it crosses the membrane. Okay, so it's got, if you count those sections in the membrane, 1, 2, 3, 4, 5, 6, 7. So 7 transmembrane domains. This one happens to have 4 extracellular domains and 4 intracellular domains. Just ways of describing it. It's not very exciting, but once you know how to do it, it's easy. Okay, so this is a little bit scary, but don't worry about it too much. We'll go over it. And also, yeah, you don't need to know this stuff in detail yet, but if you go into a university course that covers it, then yes, obviously. So basically we have the receptor and it is going to come into contact with a G protein. Okay. So if we look at picture 1, we've got our receptor protein, which is the like dark blue. We've got our G protein in the middle. Okay, so the G proteins are called G proteins because, oops, I just went ahead, because they use GTP. I've just forgotten. Who did I just ask? Oh, I gotta keep track of this. As Kathy, I think. Okay, so Sophia, do you know what GTP is?
No.
No. Emily, do you know what ATP is?
No.
No. Oh man, who was sorry? No, I don't know who was teaching you before. It's fine. Okay, so ATP is adenosine triphosphate. It is the main energy molecule of the cell. So basically, anytime the cell wants to get anything done, it has to spend ATP. So it's a specific molecule. It looks like this. So it's an adenine essentially attached to 3 phosphate molecules. Okay. So that's our ATP. And the way you get energy is you break this bond. So it's an ATP, and then we're like, oh, let's turn it into an ADP, adenosine diphosphate, because we went from 3 to 2, so tri to di. So we go from ATP to ADP, and that gives us some energy. So it's like spending money, but energy, not money. So there's another molecule that's slightly different shape. It's a guanine attached to 3 phosphates. So it's called a GTP, what happens when we break this bond? What do you think we call that molecule now, Kathy? So it was called a GTP, so guanosine triphosphate. What if now it only has 2? No? Okay. So it's called a GDP, same thing. So guanosine triphosphate, 3 phosphates, guanosine diphosphate, 2, right? Di. This is going to come up again. I'm assuming you guys want to be doctors. This will come up again and again for the end of time. You will never be able to unlearn this. I'm sorry. Okay, so basically, that's what's happening here is it's an energy molecule and it can activate things or inactivate things. So our dark blue G protein coupled receptor, it wants to get something done. So it needs our G protein. But in step 1,